so thanks everyone for being flexible. I'm really excited to get to tell you about stuff we're doing in the lab on the topic of high throughput approaches to study protein stability, design, and dynamics. So just at the highest level, there's basically three themes of our work. The first theme is to use really simple proteins that are like 40 to 80 amino acids long, protein domains, as model systems. And one of the great things about using these small domains, they're easy to model, and we can also synthesize the genes encoding these domains, like hundreds of thousands of different sequences at pretty low cost. So the next theme is to experimentally measure the properties of these simple proteins, measure their stability, their dynamics, or their function, again, at a really large scale, like from 1,000 proteins up to 1 million proteins. And then the last theme is to take all this data and translate it into computational models that people can use to understand biology and to design better proteins. And so all these three themes will kind of come in and out of the different projects that I'm going to tell you about. So we're working on projects in a few different areas. The first is to just try to learn to design really, really stable proteins, not necessarily proteins that have functions, but just fold into incredibly stable structures. And then one of the things that we want to do is to take those structures and modify them so that they can be inhibitors or therapeutic candidates, like to block protein-protein interactions. Another area that we're really interested in is designing proteins that can get inside cells, possibly by being endocytosed and escaping out of endosomes, and then interact with intracellular drug targets. And the last major area is to understand folding and conformational dynamics of proteins, again, trying to learn from these simple model systems. So I'm going to talk mostly about two projects, but if there's time, I'll just really quickly touch on a third. The first project I'm going to talk about is our work to understand how a protein sequence determines its folding stability. The second thing I'm going to talk about is how that sequence determines conformational fluctuations within the folded state. And this project is really in the preliminary stage, but I might just touch a little bit on what our strategy is to try to understand how to deliver mini protein therapeutics into the cell cytosol. So first, on the topic of folding stability, we're interested in protein folding stability for two main reasons. Oh, and they're going to appear out of order. The first reason is to understand how, so the second reason is to understand how to take a protein sequence in a genome or a variant, like you sequence someone's genome and you see all these variant proteins, and to predict what the phenotypes of those proteins are going to be. Because if you change the protein stability, that's going to have all these downstream effects on how much it aggregates, or can it perform its function, or how quickly does it get, what's the abundance inside the cell. So all these things are tied to stability, and we'd like to be able to predict that from sequence. And then the reason that I meant to mention first is that we want to learn how to design really high stability proteins. And this is actually still really hard, believe it or not. Lots of times we design something that looks really good on the computer, and maybe we predict its structure with a tool like AlphaFold, and that structure is predicted to be exactly how we designed it. But then when we experimentally test that protein, it doesn't fold at all. And so we need to learn how to predict stability from sequence so that we can solve this problem. Now, I think that the lack of data is the major bottleneck to predicting stability. So, many of you are familiar with the AlphaFold model, and AlphaFold was just this tremendous advance in predicting a protein's three-dimensional structure, but it's not able to predict the thermodynamic stability, the balance between folded and unfolded, because there isn't any of that data that went into training AlphaFold. But what AlphaFold does show is the potential of machine learning in protein modeling in cases where there's large amounts of data. Now, lots of people have certainly tried taking the data that we have, that people have measured over many decades on protein stability, and using that to build lots of models to predict stability. like every month you see someone's new model on taking the same 10,000 stability measurements and building a new tool to predict that based on structure. But there's lots of problems with this current small data set of folding stabilities that I don't want to go into the details of, but I like to compare it to like criticizing a restaurant for saying that, oh, the food there is terrible and the portions are really small. So like I said, the existing databases of protein stabilities they're small, there's lots of bias in what types of sequences are present. People use different conditions from lab to lab, and those conditions can have large effects on measuring stability. But because it's really important to try to understand stability, people still use this data to make all these different models, even though there hasn't been much progress over time. And so we really need new methods to collect better stability data. So the approach that we're taking in our lab is to develop new technology to measure millions of folding stabilities. And this is the work of postdoc Kotaro Tsubayama that I think we're going to have a first preprint on in the next month or so. So I'm just giving the high-level overview of this project. But basically, our strategy for measuring stability is to use a fully cell-free method based on a selection technology called cDNA display that was originally developed in 2009, and then Kotaro adapted it to measure folding stability. So the way we do this is we take all the sequences of the proteins that we want to measure, and we've done this with libraries up to actually 900,000 sequences was our largest library, although I wrote 800,000 here. And then each different sequence gets that DNA synthesized as an oligo. The whole pool is synthesized together at pretty low cost. And so each oligo is 300 nucleotides long and encodes a completely customized sequence of whatever we want, as long as it fits within 300 nucleotides. And then we take all those oligos and we mix them with polymerases and ribosomes in a cell-free protein synthesis system using the special technology of cDNA display, so that at the end of translation, every protein is linked to its mRNA, and then we actually reverse transcribe that mRNA into cDNA so that there's a covalent attachment between the cDNA and the protein. So now if we want to know what this protein is, all we have to do is sequence the cDNA and we can find out what it is. And then at the N terminus of each protein is a epitope tag that binds to an antibody. So we have our tag and we have our protein, and on the C terminus is the cDNA. So then we mix all these proteins with proteases, and basically the proteases can only cut the proteins when they're unfolded. And it doesn't even matter what these proteins are. We're using semi-specific proteases like trypsin and chymotrypsin that will basically cut anything when it's unfolded. But basically, because the proteins have to unfold in order to get cut, by looking at how fast they get cut, we can measure the equilibrium between folded and unfolded. And so the final way that we do this is by pulling down our proteins with beads with antibodies that bind that PA tag. So all the PA tags get pulled down, but the nucleic acid will only come along if the protein is still intact. So then by sequencing these nucleic acids, we can see how much of all these proteins survived this protease challenge, and we can compare that to our starting sample to learn how protease resistant all these proteins are. And one of the nicest things we found with this experiment is that for small domains like what we're looking at, this actually gives a very quantitative measure of folding stability. So what I'm showing you here are about 1,000 different protein variants, where these are all single point mutants of the domain protein GB1. It's a 56 amino acid domain. And on the x-axis are the stability measurements from Kotaro's high-throughput assay that measures all of these together by sequencing. And on the y-axis, this is a paper published in 2019 where they used a robotic system to actually individually purify every single one of these mutants and do a traditional stability assay with a fluorescence readout and different amounts of denaturing. And you can see that the correlation between our high throughput data and measuring these proteins one at a time is really, really good. And there's these outliers, but actually, I think these outliers, I think our data is correct on these outliers and not these experiments, but I'm not going to go into that. But basically, it's a very good correlation. Not just correlation, but actually, you know, quantitative agreement between what we measure and what they measure. There's actually a little bit different in conditions, so there's an offset. But anyway, our data were measured in a library containing 700,000 different sequences. So the 1,000 sequences shown here were just like a drop in the bucket of everything that was measured in this experiment. And the data wasn't just nice for this one protein, but it was nice for all the proteins that we looked at where there was previous stability data on mutants of that protein. So this is 10 different proteins, and these proteins have like 20 to 100 different mutants each. And we compare our high throughput data on the x-axis versus the individual experiments from the last few decades of folding stability on the y-axis, and there's really nice agreement. So we think this data is going to be really, really useful for building predictive model of folding stability. And just to give a kind of high-level summary of what we've done with this approach, today we've measured about 3 million folding stabilities, and unlike the existing data, all our folding stabilities are measured under identical conditions. So we don't have these folding stabilities measured in this buffer and these stabilities at that temperature. It's all consistent, and that's going to be really helpful for machine learning. In these experiments, we took about 250 natural domains and 200 design domains and did what's called deep mutational scanning, where we look at every possible single mutant at every different position. And unlike other experiments, we also included insertions and deletions into these libraries, which have really never been studied on any kind of scale. We also assayed about 150,000 double mutants to help use this data to understand interactions between residues. We also measured stability for about 300,000 sequences from metagenomic databases, where we just cut out single domains that were short enough to encode on the oligos from all this metagenomic data. So this is basically much more diverse sequences than the mutational scanning, where we're just looking at small variants of the same domains. So basically we have kind of in-depth data where we take one sequence and make all these mutations. And we also have breadth data where we look at all these diverse sequences from metagenomics. And the goal is to take all this data and integrate it into an AlphaFold-like model to predict structure and stability together. So the way that people have always predicted stability before is basically you build a structural model of your thing, and then you analyze the structure to predict the stability. But the approach that we're trying to take is to basically predict structure and stability in parallel. And you could ask me after the talk, but I think that will solve a lot of problems with the old approach, where first you make the structure and then you try to calculate the stability. Okay, so jumping to the second part of the talk, I want to go, the goal of this project is to study proteins in even greater detail than looking at global folding stability. So the idea of global folding stability is that we're simplifying a protein's entire energy landscape down to just the folded state and the unfolded state. But there's proteins do lots of things even within the folded state, and we call these conformational fluctuations. And fluctuations have been really hard to study in a high throughput way in the past, but they're really important. And I'm just going to give 3 quick examples of cases where fluctuations are really important. The first is a paper from 2005 that was studying mutants in the protein human lysozyme. And so human lysozyme is just an incredibly stable protein relative to like the typical protein. And these mutants, they destabilize the protein somewhat, but even the mutated proteins are still incredibly, incredibly stable in terms of their global stability. But what these mutants do is they cause a particular region of lysozyme to have conformational fluctuations, like local unfolding, that doesn't happen in the native protein. And these local unfoldings actually seed the formation of amyloid fibrils that basically cause these big amyloids to develop in the liver and the kidneys and kill the people with this inherited disorder. And it's not because the whole protein is unfolding, it's just because there's local fluctuations in a particular domain that can cause amyloid formation. Not even a particular domain, a particular like small chunk of the folded domain that cause aggregation, amyloid formation. Another example is studying aggregation of an antibody fragment. So, you know, antibodies make up a large amount of protein therapeutics, and they're a really useful therapeutic modality. But when you formulate an antibody therapeutic, oftentimes you're packing a huge amount of protein into like a really concentrated solution in a syringe, and you have to make sure that that antibody doesn't aggregate. And this was a study studying the aggregation of the antibody Fab domain. And what they found was that this particular Fab domain could kind of populate an expanded conformation that was different from unfolding. It was kind of a conformational change, but this expanded conformation was critical to the mechanism of aggregation of this domain. So again, it's aggregation caused by conformational fluctuations and not actually unfolding. And then the last example is really interesting. This is a case of people that were designing an HIV vaccine candidate where they had a protein that produces a particular conformational epitope. But what they found was that their designed protein didn't always stay in exactly the same conformation. It could fluctuate and expose surfaces that were not exposed in the normal native structure. But basically, these alternative states caused other types of antibodies to form, antibodies that were not actually neutralizing. So basically, their vaccine created, different types of antibodies, and some of the antibodies weren't helpful, and those were the ones that were being triggered by this conformational fluctuation. So in this particular paper, they actually created mutations to minimize that conformational fluctuation and promote the formation of the good antibodies, the ones that would actually be neutralizing. So again, they weren't seeing unfolding, they were just seeing small conformational fluctuations. And so these are all examples of the importance of conformational fluctuations in biology, in therapeutic formulation, in protein design. And unfortunately, these conformations are really difficult to predict. So the goal of this project is to develop fast, accurate models to predict the energies of conformational fluctuations for any protein. And we can't do this right now. People can run simulations of proteins and maybe see which segments are flexible and which segments are not. But I don't think there's a lot of validation of that approach. And so I think that a data-driven approach where we measure fluctuations for thousands of different proteins and then learn from that data rather than kind of a first principles modeling approach, it will at least be complementary, and I think it's going to really help this problem of predicting fluctuations. So this project is being led in the lab by 3 postdocs, Sugi, Alan, and Jane. And just like before, our strategy is to measure conformational fluctuations for thousands of small domains, except now, instead of using DNA sequencing, we're using mass spectrometry, and specifically the method called hydrogen exchange mass spectrometry. So, many of you are probably familiar with hydrogen exchange. If you're not, I'm just going to quickly describe it. Hydrogen exchange works by taking a protein that's in water and then diluting that protein into D2O. And so when you put your protein from water into D2O, the first thing that happens is all the backbone amides that were on the surface of the protein, their hydrogens come off and they get replaced by deuteriums. So you get deuteriums on any amide hydrogen that was exposed to solvent. But the places in the protein that are in secondary structure, those stay as hydrogens. Now, eventually these hydrogens will exchange to deuterium, but that only happens when the protein undergoes some kind of conformational fluctuation that opens up those pieces of secondary structure so that now the hydrogens can come off and the deuteriums can come on. And so basically, if you look at the rates of these exchanges, then you can learn, depending on how you do the experiment, you can either learn the kinetics of these fluctuations, or you can learn the thermodynamics of these fluctuations. And we're interested in the thermodynamics. So anyway, yeah, I'll quickly just show this one example protein. So this is a really simple domain. This is a domain that I designed, actually. It has only 40 residues, and each different position is colored by the exchange rate at that position. Now, you know, for residues on the surface, they exchange really fast even when they're folded. So residues on the surface don't tell you anything about folding or unfolding because they're just going to exchange really fast no matter what. So we don't worry about those as much. But now I'm putting the actual numbers up on this protein, and just focus on this top helix. These are all residues that form hydrogen bonds in the helix, so they exchange slowly. And so right here in kind of the core of the protein, this residue exchanges with a free energy of about 7.5 kcal per mole. So that almost means that like 1 molecule out of 1 million is open and exchanging at any given time. And then down here on the other side of this helix, this residue exchanges at an energy of 3.5 kcal per mole. So a little less than like 1 molecule in 1,000 is open at any given time. So you know, even in this really small, simple protein, there's a big difference in conformational fluctuations at this site in the core of the protein and this site on the other side of the same helix. And so we'd like to understand how to predict these opening energies based on the sequence. And so we need to collect a lot of data. So the approach that we're using, once again, uses oligo pools, except instead of attaching the protein to the DNA and then reading out the DNA, we're just using the oligo pools to make mixtures of soluble proteins. So we assemble a library of a bunch of domains that we want to measure, and then we encode all those sequences in oligos, and then we take all of those oligos, put them into a vector, transform them into E. coli, and grow it all in the same culture. And then we purify out the protein mixture. So this is basically exactly the way that you purify one protein, except we're purifying, since we started with a DNA mixture, we purify out a protein mixture. You know, we purify it away from all the other stuff inside the E. coli. And actually, because of what we have a way of doing this so that everything kind of expresses in about the same amount. Some things don't express, but we get mixtures that contain most of the proteins that we put into them. And then to measure the hydrogen deuterium exchange, we measure this by mass spectrometry. So we take our whole mixture, we put it into deuterium for 5 minutes, and then we quench that reaction so the exchange stops. And then we inject that mixture into LC-MS, which separates out all the proteins and measures their masses. So for each different protein, we get data that looks like this. Like this is the protein's mass distribution before it's seen any deuterium. And then after 5 minutes in deuterium, it gets a little heavier. That's one LC-MS injection. And then we have a different LC-MS injection where all the proteins were in deuterium for 10 minutes. We have another LC-MS injection where all the proteins were in deuterium for an hour. And so each protein, we basically watch how it gets heavier at all these different amounts of time in deuterium. So this is just one way of visualizing all the different proteins in our sample. So each protein elutes from the LC-MS at some different time, and all the proteins have different masses. And these masses change a little bit, but they don't change a lot. And then we also have another dimension of separation in our LC-MS experiments called ion mobility. And so all the different proteins here are colored by their ion mobility drift time. But basically, each one of these circles is a different signal. And what's important is when the proteins pick up mass in these HD exchange experiments, their LC retention time stays the same, and their ion mobility drift time stays the same. So we always know that this circle is the same protein, even though its mass is like getting heavier with each different injection when it's been in different amounts of deuterium. So this is what lets us monitor a huge number of proteins in one experiment. And now you might be familiar with other applications of hydrogen exchange mass spec, where people take a protein and they break it up into a bunch of peptides. In order for us to look at as many proteins as we can, we're not actually doing that right now. We're just looking at each intact protein. And so, that doesn't give us the same resolution as if we broke each protein up into peptides, but it lets us pack a large number of proteins into our sample so we can learn this relationship between sequence and conformational fluctuations. So this is the way that we look at the data. This is a particular protein fold that has about 40 residues. And what I'm showing here are 400 examples of different protein sequences that we know more or less all fold into this same fold. They're not like one mutation away from each other. The sequences are quite different. They have maybe 20% or 30% sequence identity to each other, but they all have about the same fold. And what each curve are, each curve shows the distribution of opening energies in that particular protein. So, we look at each protein get heavier over time, and when we see that, we don't actually know which residue is exchanging. So like, we don't know that residue 15 is really stable and residue 25 exchanges really fast. We just know the overall distribution of opening energies for all the different residues. But I think these distributions are really fascinating. So like, just starting from the bottom, there's this purple protein that basically has the opening energies are very low for all the residues. So basically all the residues exchange really fast, and this protein probably doesn't really fold correctly. Then at the top, there's a really stable protein like this green protein, but the green protein is kind of interesting because it has about 15 residues that are really, really stable. We don't know where those 15 residues are, but we know the number. They're all stable between like 5 and 6 kcal per mole. And then it drops down by quite a lot. And then the remaining residues are much less stable. And that's very different from the red protein that again has about 15 highly stable residues, but then the rest of the red protein is substantially more stable than the other residues in the green protein. And that's really obvious between the orange and the blue. So like 35 of the residues in the orange protein all open at about the same energy. But in the blue protein, there's this huge difference between the 18 most stable residues and the remaining 20 residues in that protein. So every one of these patterns is different. And there's also all these gray lines back here from the 395 other proteins in this fold. So I think learning from this data is going to be really fascinating to understand why the different sequences of all these different proteins lead to these really different energy landscapes in terms of which ones have low energy conformational fluctuations and which ones don't. Actually, so I'm showing 400 landscapes right now, but the postdocs in the lab have actually successfully measured landscapes for about 7,000 proteins, small domains like this. And we're just starting to take all that data and trying to learn what's going on. I'm going to show one quick example of where we used NMR to dive more into the details of one of these proteins. So here I'm showing 2 different proteins that have that characteristic that I showed before, where lots of residues in the red protein have a kind of similar opening energies. But in the blue protein, there's like 16 residues that are high stability. And then there's another 20 residues that are much lower stability than the comparable residues in the red protein. And so we used NMR, where we can actually see the exchange rate of every different residue to understand what was going on with these 2 proteins. And so these 2 proteins both form 3 helical bundles. So actually we solved the NMR structures of these 2 proteins. So we know exactly how they fold. They both fold into these 3 helical proteins just as they were designed to. They're a little bit different in the orientation of helix 1. And so here I'm showing the opening energy at each residue from the N terminus to the C terminus. Now because we're doing NMR, we know which position is, we know which energy is which residue. So in helix 1, the blue protein is more stable than the red protein. And in helix 2, the blue protein is again more stable than the red protein. But in helix 3, the red protein, helix 3, is basically the same stability as the other two helices, but helix 3 in the blue protein is way less stable. It actually exchanges so fast that we couldn't really measure the opening energies except for these two residues. Now, I want to be clear that helix 3 is not unfolded. If we look at the native structure, we see that it's folded exactly as designed. It's just that 10% of the time or something, or 5% of the time, it's fluctuating and opening up in a way that the comparable helix in protein 1 doesn't do. And somehow the other two helices are actually more stable, even though this third helix is like coming on and off. So I can't tell you why the sequence of protein 2 causes these fluctuations, but we have this really exciting data set of about 7,000 proteins, and I hope we're going to learn some answers to these questions. Okay, I'm going to skip the last part of the talk on delivering mini protein therapeutics, but if you're interested in this, I can show you the slides if you ask a question about it. Okay, so I'll skip through this, and I just want to thank the people in the lab, especially Kotaro, who pioneered the cDNA display method for measuring global stability, and then Sugi, Alan, and Jane, who are really pushing forward the HDX method. Yeah, thank you, and I'm happy for your questions and the opportunity to speak. Thanks.